To my molecular biochemistry series. In this episode, we are focused on DNA repair mechanisms. In the previous episode, we discussed a lot about DNA mutations. And so we want to think about how can we repair a DNA strand or helix that has a mutation, or if it just has damaged nucleotides or damaged bases of those nucleotides. And so that is where we are going to continue our discussion. And the first repair mechanism is the nucleotide excision repair mechanism. And as its title suggests, we are excising out a damaged nucleotide or more than one damaged nucleotide. And to do this, an endonuclease, an enzyme, steps in and cuts out the damaged nucleotides. Also, fully functional, non-damaged nucleotides get caught up in this reaction that cleaves the backbone and removes this section, and then DNA polymerase will come in and replace those damaged nucleotides with fully functional ones, and ligase will seal up the cut in the phosphate backbone, and voila, we have a fully repaired DNA single strand that was previously damaged. Now, nucleotides can be damaged by free radicals or even the sun's radiation, so UV light exposure, especially prolonged exposure. And one disease is xeroderma pigmentosum, where if you're unable to repair the breaks by these free radicals or UV light, you can develop some symptoms such as dry skin, or you become very photosensitive. In addition, you could even be more susceptible to skin cancer if your cells are not able to properly repair these damaged nucleotides. The next repair mechanism we can look at is base excision repair, where, just as its title suggests, we are excising the base of a nucleotide. And this is a much more precise mechanism. It is more meticulous, and it relies on a endonuclease as well to cut the 5' end, but differently than nucleotide excision repair, we need a lyase to cut the 3' end of the phosphate backbone. And so this will allow the glycosylase to step in and enzymatically catalyze the removal of the damaged base of the nucleotide. And so we can remove that, replace it with a proper base, and polymerase will come in and return the nucleotide to the DNA strand, and ligase once again seals the backbone. It might also be worth mentioning at this point that an endonuclease is able to jump in really wherever it wants within the DNA backbone, while an exonuclease has to move from either I can't recall if it's a 3' or 5' end, but it has to operate from the end of the DNA strand and move to the other end. So it operates more of its capacity on the ends of the DNA, while a endonuclease will operate more within the DNA. The next repair mechanism I'd like to discuss is the mismatch repair mechanism, and this is highly relevant to the DNA mutations I was discussing in the previous video, where you could have a nucleotide that is mutated to a different nucleotide. And if this occurs, especially on the template strand, our pre-mRNA is going to have a different nucleotide, and this is going to tell the ribosome to translate a different amino acid and thus cause the protein to have a different structure, and it could cause it to misfold, become non-functional, and thus the cell will not have the operation that it needs, and can damage our cells and tissues, causing disease or disorder. And so this mismatch repair mechanism is important, and it often occurs. This mismatch can occur due to DNA replication errors. If you recall from my video on DNA replication, there is a lot of moving parts, a lot of enzymes catalyzing a lot of different reactions, and so errors are going to occur. And hopefully they don't accumulate, because our mismatch repair mechanism is stepping in and slowing the accumulation of those errors. And similar to the other mechanisms, an endonuclease will step in and slice out a section of the helix that has the improper nucleotide with its counterpart, and polymerase will step in and replace it with the proper ones. The other concept to discuss is if there's double-stranded DNA break, thus nucleotides are lost, there is a mechanism called homologous recombination and non-homologous recombination that will repair this break in the double-stranded DNA. And ionization can cause double-stranded DNA breaks, charged particles moving through and reacting with the phosphate backbones and causing it to fragment. And so homologous recombination relies on another DNA helix to come in, serve as a template strand, and polymerase can step in and provide the proper nucleotides based on that template strand. However, in non-homologous recombination, those nucleotides that were lost are lost permanently, and the fragments are just brought together, and a shorter DNA helix is going to result. And this will most likely code for different proteins, and so this repair mechanism is not as effective as homologous recombination. But at least it's something so that the DNA isn't completely unable to code for or inform the cell on what proteins to make. That is a great overview of DNA repair mechanisms, and I'll see you guys in the next one. All right, bye.